Hey guys, today we will be focusing on eukaryotic gene regulation. So last time we talked about prokaryotes, now we're going to look into eukaryotic gene regulation. So this is a question that should have been covered last week. We did go over it a little bit, but I just want to make sure that I was clear on it. So enhancers are said to be cis-acting. What is meant by cis-acting and what are enhancers? So cis-acting means that genes under control must be in the same chromosome as the cis-acting element. So when you have something cis-acting, it's within the same chromosome. So genes under the control of this guy is stuck to the same chromosome as it. Enhancers are sections of DNA that regulate transcription of other sections of DNA. So if you have a enhancer, it just kind of, so enhancers kind of mobilize everything going on. It can be located upstream, downstream, or within the gene that it regulates. Okay. It is orientation independent and can act at large distances from the promoter. So if we have a promoter in here somewhere, or that promoter, so we have the promoter here. If that promoter pushes it off, then you can even do it within a whole lot of distance between each other. It can still occur. And it binds special transcription factors that actually increase the transcription rate. Okay. So enhancers just bring different transcription factors that actually increase the rate of transcription and allow you to transcribe even faster. So what do you guys remember about the eukaryotic gene components? We have exons, which are the coding regions, introns that are the non-coding regions, the transcription start site, then we have the promoter. In the promoter, in a eukaryotic gene, we have a core promoter. This involves the Tata box. This thing will come hunt you forever. I swear, the Tata box keeps following me. Anyways, it is common. This core promoter is very common among most eukaryotic genes. It also brings in a transcription factor called transcription factor 2D, which is essentially near the Tata binding domain. And then we have transcription factor 2B or TF2B. And this, and so these components of the core promoter. Then we have the proximal promoter elements, which involve segments of DNA found upstream. So very upstream from, so we have this guy here, then we have the proximal promoter elements, which are upstream of the core promoter. So we have the core promoter here, and then everything else is kind of nearing it. And then it varies from gene to gene. So that's kind of how it varies. Then we have distal control elements, which essentially brings in enhancers and silencers and all that fun stuff that actually is involved with regulating gene expression. So one of the major systems in eukaryotic gene regulation is the GAL gene system. The GAL gene system in yeast contains 4 structural genes: GAL1, GAL10, GAL2, and GAL7, right? And then we have 3 regulatory genes, which involve GAL4, GAL80, and then GAL3. The products of structural genes, so the products of this guy transport galactose into the cell for metabolism. The positive control for this system involves the activator protein. So you need the activator protein to be present to turn on the gene transcription. In this case, specifically, GAL1 and GAL10 genes are both positively upregulated. It is controlled by the UASG strain sequence region, control region, which is essentially standing for upstream activating sequence of GAL genes. So U is upstream, A is activating, S is sequence, and the G is just saying that it is of the GAL genes. Okay. This involves 4 binding sites for GAL4 protein, right? So essentially 1, 2, 3, 4. UAS is functionally similar to enhancers in multicellular eukaryotes. So this system is specifically in not specifically in yeast, but this is one of the systems that were originally found in yeast. And this region, this control region, is found in is functionally similar to various enhancers in multicellular eukaryotes. Again, GAL genes are inducible. Transcription is regulated by the presence or absence of galactose. So when you think GAL genes, think of processing galactose for metabolism. It brings it in, it processes it, and it's happy cells. Absence of galactose in this case is you see there is no transcription of the GAL structural genes, okay? And if galactose is added to the medium, transcription ends up starting immediately because it is inducible. So when galactose is present, it goes right into transcription mode. Like, no stopping me now, okay? So GAL4 protein. It is encoded by the GAL4 gene. It is negatively regulated by GAL80. So we have an GAL4 here, this guy over here. It's binding to UAS, having fun. And then GAL80 here is coming in, keeping it inactive. But then when galactose comes in and GAL80 is like, oh no, this is trouble. I'm leaving. Bye-bye. And then it starts activating the GAL4. So in this case, the GAL4 protein has a DNA binding domain that recognizes and binds to the UASG region. So as I said, it binds here. Binding of phosphorylated galactose to GAL80P or GAL4P exposes activation of the activation domain of GALP4. So it kind of opens it up. It's like, oh, I need to get things done. Come on, Come on, guys, we need to get started. And it essentially recruits everything that you need to start transcription, okay? So the next question here that we're going to talk about is explain how the following mutations would affect the transcription of the yeast gal1 gene. A deletion within the gal4 gene that removes the region encoding amino acids 1 to 100. So what would happen? How would this affect transcription of gal1 genes? So a deletion within the gal4 gene that removes amino acids 1 to 100 would remove DNA binding sections and not allow transcriptional activation. So essentially, this ends up removing that DNA binding section, or the UAS region. It takes it out. It's like, no transcriptional activation whatsoever. Are we clear? Okay. That's what happens if a deletion within the gal4 gene occurs. If it removes this many amino acids of it. What happens if the entire gal3 gene gets deleted? Without the product of gal3, there would be no disruption of the gal4 gal ADP complex, and therefore no transcription of gal1 gene would occur, right? Because remember, if gal3 comes into play, there is no disruption of that. So it keeps it. So remember, gal80 inhibits gal4 from activating transcription. Gal3 comes in, the product of gal3 comes in and disrupts this complex to actually start transcription. So if gal3 was completely deleted out, it wouldn't disrupt that complex and then end up with no transcription whatsoever with gal1. Okay? So a mutation within gal80 gene that blocks the ability of gal80 protein to interact with gal3p. So what happens if a mutation comes in gal80 that blocks its ability to interact with gal3p? If the gal80 gene product can't interact with gal3p, there can be no interaction with gal4p, gal80p complex, so there wouldn't be any gal1 transcription. So if it blocks the ability of gal80 to bind with or interact with gal3p, it's the same thing. It cannot interact with that complex, which essentially brings back to this situation where there would be no gal1 transcription, okay? Explain how the presence of glucose represses the gal structural genes. So how does this repress it? Glucose represses the gal structural genes by repressing gal4 transcription. Essentially, if you have a high glucose concentration, the cell activates a repression complex comprised of MIG1, MIG2, MIG1 and MIG2, DNA binding protein. So these two are DNA binding proteins that bind to a region upstream of the gal4 gene. general repression complexes that are composed of proteins TUP1 and SSN6. So you guys have, it has MIG1, TUP1, all those guys come in, SNF, all those come in, they kind of end up, and then the repression complex binds to the regulatory region of the GAL4 gene. Sorry, this is gene, not gen. Okay. So what are the three states of the GAL structural genes? The first one is repressed. And in the presence of glucose, the repressors MIG1 and MIG2 are activated and prevent GAL4 transcription. So MIG1 and MIG2 are repressors. When they are activated, it prevents GAL4 transcription. And that results in no transcriptional activators for the GAL genes in the cell, and very little transcription ends up occurring. Then you have another state called de-repressed. In this occurs in the presence of glucose or galactose. The GAL4 gene is transcribed, but GAL80 prevents transcriptional activation of the GAL genes. Under these conditions, some transcription actually does occur because the GAL4 gene itself is present. Then you have induced in the presence of galactose and the absence of glucose. So essentially, remember GAL4, it loves to be transcribed, especially when you have galactose. And when galactose comes in, GAL4 is not inhibited whatsoever. It results in a very strong transcriptional activation of these GAL genes, okay? So this is another question that I found that was in the question set that Dr. Bruce put up, except I took out the figure to it. So this is just a general question at this point. Certain mutations decrease the relative transcription rate of the beta globin gene. Where are these mutations located and how do they exert their effects on transcription? These mutations are located within the proximal promoter region and promoter elements. These proximal promoter elements are located upstream of the coding region and serve as binding sites for RNA polymerase III and general transcription factors. Mutations in these regions end up hindering protein and DNA interactions and thereby actually reduce transcription itself. Especially even if a single nucleotide is changed, the reduction in transcription activity is actually can be very substantial. You would think with one change in a single nucleotide can actually like bring it down a whole lot, like very substantially. So that's how these mutations end up decreasing transcription rates, okay? How does GAL4 regulate 4 different GAL genes at the same time? Contrast this mechanism with how or lac repressor controls the expression of 3 genes. So we know that GAL4 has a binding DNA binding domain that targets the UAS sequence, right? So this sequence is essentially present in the enhancer region of 4 different GAL genes. And we also know that GAL4 is able to activate transcription from the 4 separate GAL promoters through its capacity to bind with all 4 UASs. So all of these guys can just bind to the 4 different promoters. But in prokaryotes with the lac repressor, it controls the expression of 3 genes by blocking or unblocking the RNA polymerase to a single promoter. So in the GAL4 system, you're thinking about 4 different promoters, whereas in the the lacopron and all that good stuff, remember those guys? It's under 1 promoter, right? So that's the differences between those 2. These 2 regulatory systems are similar in that genes involved in metabolic pathways for a specific sugar are coordinately induced by the presence of the sugar. So whether it's galactose or lactose, whichever one is present, they end up breaking it down by the state of the levels of the sugar itself that they process. So the next question we have is contrast the states of genes in bacteria and eukaryotes with respect to gene activation. In general, the ground state for a bacteria gene is on. So transcription initiation is prevented or reduced if the binding of RNA polymerase is blocked. Whereas with eukaryotes, the ground state is off. So in natural settings, eukaryotic genes are typically off. So essentially, it needs the transcriptional machinery. The machinery itself cannot bind to the promoter in the absence of like an activator protein or some kind of regulatory protein that actually allows you to move forward with it, okay? Next, we have this question where it's asking, how is the activation of the GAL gene prevented in the presence of galactose and glucose? So as we've talked before, in the presence of both galactose and glucose, the activation of GAL1 is prevented by the MIG1 protein. As we remember, it is a sequence-specific DNA binding protein. So MIG1 is a sequence specific binding protein that binds to a site between the UAS element and the promoter of the GAL1 gene. Okay. And this MIG1 ends up recruiting everything like TUP1 and all that good stuff that contains a histone deacetylase that ends up repressing the gene transcription in that case. So make sure you guys definitely do understand how this system works and how it ends up getting repressed or activated, and so on and so forth. Okay. So this is pretty much the last discussion lecture you guys will have before your final. So let me know if you guys have any questions, and I wish you guys the very best and very much good luck with the rest of your semester and all your other courses. And keep in touch. Alrighty. See you, folks.